Auxin, the first plant hormone, was discovered by Fritz Wendt as a growth promoting chemical reptiles, Avena sativa, because the chemical isolated by Wendt promoted the elongation of coleoptile derived from the Greek word auxin meaning to increase. Various naturally occurring auxins are known, namely IAA, indole 3 acetate, indole 3 butyric acid, and PPA, phenyl acetic acid. Indole 3 acetic acid is the principal naturally occurring auxin in higher plants. There are also more than 200 analogues with different chemical structures known to have auxin activity. The only common features shared by these compounds are an unsaturated planar aromatic ring structure and a carboxyl side chain. Biosynthesis and transport. Major primary sites for IAA synthesis are the shoot apical meristem, young leaves, developing fruits and seeds. Intracellularly, IAA is found in the chloroplast as well as in the cytosol. In plants, IAA occurs in both conjugated and free forms. It has been found to be conjugated to both high, such as glycoproteins, and low molecular weight compounds, such as glucose. IAA conjugates are located exclusively in the cytosol. There are two pathways for the biosynthesis of IAA. These pathways are tryptophan dependent as well as tryptophan independent. Tryptophan dependent pathways, IAA is structurally related to the amino acid tryptophan. Considerable research has shown that tryptophan is precursor of auxin biosynthesis. Tryptophan is converted into IAA by several pathways. The indole 3 pyruvic acid pathway is the principal IAA biosynthetic pathway in plants. Overall, the conversion of tryptophan to IAA through this pathway involves deamination of tryptophan catalyzed by transaminase, decarboxylation of indole 3 pyruvic acid catalyzed by indole 3 pyruvic acid decarboxylase, oxidation of indole 3 acetaldehyde catalyzed by indole 3 acetaldehyde dehydrogenase. Tryptophan dependent indole 3 pyruvic acid pathway of IAA biosynthesis, tryptophan to indole 3 pyruvic acid, then to indole 3 acetaldehyde, then to indole 3 acetic acid. Tryptophan independent pathway. In addition to tryptophan dependent pathway, recent genetic studies have provided evidence that plants can synthesize IAA via one or more tryptophan independent pathways. This route does not involve tryptophan directly as a precursor to the formation of auxin. A tryptophan independent pathway of IAA synthesis starting with indole has been partially characterized in several plant species. However, the precise pathway for tryptophan independent IAA synthesis is not known. Transport. In plants, there are two distinct pathways to transport auxin, that is IAA. One is rapid long distance source to sink non-polar transport. The other is slower short distance polar transport. Polar transport occur in a cell-to-cell manner and depends on specific influx and efflux carrier proteins. Auxin is the only plant hormone known to be transported polarly unidirectionally. Polar transport of auxin is an energy dependent and a gravity independent process. Long distance source to sink non-polar long distance source to sink non-polar transport of auxin occur through phloem and it is passive in nature. Most of auxin that are synthesized in mature leaves appear to be transported to the rest of plant non-polarly. Auxin is transported polarly in a basipetal from shoot apex to base direction in the shoot. The basipetal polar transport in stem mainly occur through vascular parenchyma tissue. Polar transport does not completely um, occur through symplast. That is, auxin exit the cell through plasma membrane. Auxin efflux diffuses across the middle lamella and re-enter the cell below through its plasma membrane. Auxin influx. Auxin influx occur by two different mechanisms: simple passive diffusion of protonated IAAH from across the plasma membrane and secondary active transport of deprotonated IAA minus anionic form via 2H plus IAA minus symporter. The protonated form of IAA is lipophilic and readily diffuses across the plasma membrane. In contrast, IAA negative anionic form is negatively charged and therefore does not cross membrane by simple diffusion because the plasma membrane H plus ATPase normally maintains the cell wall at about pH 5. About half of the oxygen in the apoplast will be in the protonated form and will diffuse passively across the plasma membrane down a concentration gradient. Once IAA enter the cytosol, which has pH approximately 7.2, nearly all of it will dissociate to the anionic form. Deprotonated IAA minus anionic form enter the cell through 2H plus IAA minus importer influx carrier that is uniformly distributed around the cell plasma membrane. A 2H plus IAA minus importer mediated secondary active uptake mechanism is responsible for oxygen accumulation. Molecular genetic studies in Arabidopsis thaliana have identified a permease type IAA influx carrier termed OX1, oxygen transporter protein 1. Now, oxygen efflux is mediated by pin oxygen efflux carrier proteins. The pin proteins named after the pin shaped inflorescence formed by the pin 1 mutant of Arabidopsis are a plant specific family of transmembrane proteins that transport the oxygen. Directional oxygen movement is associated with tissue specific distribution of pin proteins. They are asymmetrically localized on the plasma membrane of cells, and their asymmetrical distribution also determine the directionality of intercellular oxygen flow. Until now, 8 members of pin protein family have been isolated in Arabidopsis and are commonly referred to as pin 1 to 8. Pin 1, 2, 3, 4, and 7 proteins are localized at the plasma membrane, whereas where they act as oxygen efflux carrier. Pin 1 localized basally in vascular parenchyma cells mediate vertical transport of oxygen IAA from the shoot to root along the embryonic apical basal axis. Other proteins that play a role in oxygen efflux are ABCB proteins, ATP binding cassette subfamily B. The ABC transporters comprise a superfamily of membrane proteins. On the basis of sequence similarity, protein size, and other features, the plant ABC proteins can be divided into 8 subfamilies ABCA, ABCA to ABCH. Among the ABCB subfamily, ABCB1, ABCB4, and ABCB19 have been well characterized as oxygen transporters. However, recent studies show that other ABCBs like ABCB14 and ABCB15 are also associated with polar oxygen transport. ABCB19 mediates rootward oxygen transport in the steel. Many oxygen transport inhibitors phytotropins are presumed to bind the IAA efflux protein and prevent oxygen efflux. Synthetic compounds TIBA, 2,3,5 triiodobenzoic acid, and NPA, naphthyl thalamic acid, act as oxygen efflux inhibitors and thus block polar transport. In roots, on the other hand, there appear to be two transport streams: an acropital stream arriving from the shoot, flow through the xylem parenchyma cells in the central cylinder of the root, and direct oxygen toward the root tip. A basipetal stream then reverses the direction of flow, moving oxygen away from the root tip or basipetally through the epidermal cells. In the primary root, acropital transport of oxygen toward the root tip is a pin dependent process. Oxygen flow toward the tip is maintained by action of pin 1 and pin 4 proteins present in the steel. In the columella, the action of pin 3 and pin 7 redirect oxygen flow laterally to the lateral root cap and epidermis, where the apically localized pin 2 mediates the upward flow of oxygen to the elongation zone. The pin 2 based shootward epidermal oxygen flow is further supported by action of OX1 and ABCB4 transport. In the acidic cell wall around pH 7, approximately 50% of IAA is protonated. H plus ATPase help to maintain pH gradient across the plasma membrane. Protonated IAA, that is IAAH, may enter the cells by simple diffusion, while the deprotonated form may be taken up through 2H plus IAA minus importer OX1 protein located randomly on the plasma membrane. In the cytosol pH7, the deprotonated form IAA minus will dominate. IAA minus can exit the cell only through efflux carriers, pin auxin efflux carrier proteins. The uniquely basal location of efflux carrier is the key of polar transport. Polar localization of pin proteins in the Arabidopsis root tip. The directionality of auxin transport is determined by polar subcellular localization of pin proteins. Pin 1 and pin 4 are localized at the basal root apex facing side of the xylem parenchyma cell in the central cylinder of the root. Pin 2 at the apical shoot apex facing side of the epidermal and root cap cells. Pin 3 and pin 7 are localized uniformly on all the sides of columella cells of the root cap. Pin 3 and pin 7 play a central role in regulating the lateral translocation of auxin. Physiological effects of auxin. Cell elongation. Auxins stimulate cell elongation or expansion in stems and coleoptiles. According to the acid growth hypothesis, auxin promote elongation growth by increasing cell wall extensibility. Auxins stimulate proton pumping by activating directly or indirectly proton pump. H plus ATP is present on the plasma membrane. As a result, the pH of cell wall fall as low as 4.25. The low pH activate a class of cell wall protein termed expansins that disrupt the hydrogen bonding between cellulose microfibrils, causing the laminate structure of the cell wall to loosen. With the rigidity of cell wall reduced, the cell can elongate. The expansion triggered loosening of the wall is reversed when the pH is raised back to the normal, showing that expansins does not break covalent bond in cellulose like auxin. A fungal compound fusicosin induce rapid cell elongation and trigger proton pumping out of sensitive cells with the accompanying wall loosening. The action of fusicosin or auxin can be blocked by permeating the cell wall with the buffer that prevent the extracellular pH from being lowered. Cell differentiation. The differentiation of relative amount of xylem and phloem are regulated by auxin concentration. In general, high auxin concentration induce differentiation of xylem and low auxin concentration induce phloem differentiation. Rooting. Auxin inhibit elongation of primary root but stimulate root initiation on stem cuttings, that is adventitious roots, and lateral root development. Lateral roots are commonly found above the root hair zone and originate from the cells present in the pericycle. Auxins stimulate these pericycle cells to divide. The dividing cells gradually give rise to lateral root. Apical dominance. In most plants, the growing apical buds initiate the growth of lateral buds, a phenomena called apical dominance. Removal of shoot apex usually result in the growth of lateral buds. Plants with strong apical dominance are typically weakly branched and show a strong branching response to decapitation, removal of shoot tip. In the presence of apical bud, the outgrowth of lateral bud is inhibited by auxin. Auxin in the shoot apex maintains apical dominance by stimulating strigolactone synthesis. The strigolactone activate the branched 1 BRC1 gene. The product of BRC1 gene inhibit in lateral bud growth. Cytokinins antagonize the effects of strigolactones. Fruit set. Fruit set is defined as the transition of quiescent ovary to a rapidly growing young fruit. Auxin promote fruit development after fertilization. The fruit growth may depend on auxin produced in developing seeds. Auxin is produced in the endosperm and the embryo of developing seed. In some plant species, seedless fruits may be induced by the treatment of unpollinated flowers with the auxin. The production of such seedless fruits is called parthenocarpy. Tropic response. Auxin induced differential growth in plant organs and under the influence of directional stimuli, that is light and gravity. This is termed as tropic response. According to Colondi-Wendt theory, which state that tropic stimuli induce the lateral redistribution of auxin, resulting in unequal accumulation between opposing regions of responding organ and thus differential growth.